And now we will move to Adam Kepecs from the medical school in Washington University.
Well, first, what a wonderful set of talks, and I'm really honored to be included in this. It's really a fascinating new era meeting across fields. I'm especially grateful to Cold Spring Harbor, my longtime home, for this invitation. So I'm going to talk about brain-body interactions gone awry in sort of a different way than what you've heard. In the context of inflammation and apathy. So the first question I wanted to start with is that it's useful to change behavior when you're sick or injured, right? But different situations call for different responses. So if you stumble and sprain your ankle, it would be a good idea to stop moving and rest your ankle and let yourself recover. You feel that your ankle hurts because there's pain receptors and your brain interprets the neural input appropriately. So you stop moving, and this seems like a good idea. It's adaptive and again, helps you get better faster. Now, if you catch a cold, you get sick and typically feel fatigue, fever, maybe a loss of motivation and social drive. These kinds of behaviors are collectively referred to as sickness behaviors, and they're also probably adaptive. Certainly, fatigue can save you energy for fighting infections and so on. Now, the brain orchestrates all these behavioral and physiological responses too, but how does it sense pathogens? So pathogens enter the periphery, right? So the brain doesn't have direct access to them. And of course, they require receptors that the brain doesn't have. So it's the immune system that gets activated first, and its main business is to fight and get rid of the pathogens. But it also acts as a sensory organ for the brain. So it detects it detects the pathogens and releases cytokines that are detected by the brain, and again organizes this sickness behavior that are all beneficial. Now, suppose you eat rotten food. This can also make you sick, and the pathogens are sensed through an overlapping set of mechanisms. But the typical response is nausea and vomiting, which again is obviously adaptive here. But these are somewhat different responses from viral infections, although overlapping. So it's unclear how the brain could orchestrate these different kinds of responses from the signals that it receives. All right, so as a neuroscientist, sort of we think about sensory transduction sort of in the following simple scheme. We focus typically on, as neuroscientists, external sensations, how they lead to things like muscle activations based on a nociceptive neuron. In this case, and there's a sequence of events that unfolds, conveying all this information to the brain. Now, unlike for these other sensory systems studied by neuroscience, we have a rather incomplete understanding of how the brain senses pathogens and how it orchestrates the appropriate responses. So, of course, immune cells get activated first and cytokines get released. And there's at least two parallel pathways for carrying this information to the brain. Now, neuroscience frames this problem in terms of a neural code, that there is a SPI code, the rate and pattern of action potentials across different neuron types, and all this encodes features of the injury. That gets interpreted. So this is an expanding field. So I'm going to focus right now on one question today, of the two questions I'm going to mention, is what is the cytokine code, or is there a cytokine code to enable the brain to regulate sickness behavior? Now, like with any adaptive processes, of course, lots of things can go wrong. So, for instance, if there's a failure to eliminate the inflammatory inducer and resolve the infection, that will cause chronic inflammation. And there's many behaviors induced by inflammation that are adaptive, But if there's no resolution, these can turn maladaptive. And there's many chronic inflammatory conditions like cachexia, wasting syndrome, that I will discuss today. However, their unique set of symptoms, in addition to their unique set of symptoms, they also have converging behavioral symptoms. So people with chronic inflammatory conditions are more likely to have extreme fatigue, loss of motivation, apathy, loss of appetite, and so on. And vice versa, people with clinical depression are more likely to have low-grade chronic inflammation. And in fact, there's just striking similarities between these behavioral symptoms of depression and chronic inflammation, but sort of at the mechanistic level, this is not understood. And this is the second question I was going to address, a short story about how does chronic inflammation produce what we would call apathy or amotivational syndrome. And I also want to address sort of why I'm personally interested in this. Because psychiatric conditions like depression today lack valid predictive animal models. This has been really a major hurdle for the field. And we all know this, and mimicking the human disorder is just hard. How could you possibly connect the subjective feelings of a person to a mouse? So this is a topic of great interest, that something that my lab has studied, something we developed a whole approach we called computational validity, and so on, that we're pursuing, but that's not enough. What we really like to have is what you would call construct validity. Like in other fields of biomedicine, you want to accurately recapitulate the disease, which would require understanding the pathogenic mechanisms. And unfortunately, it's been challenging in psychiatric disorders like depression because it's hard to find large effect size penetrant genetic variants. So in contrast, there's numerous well-validated models of chronic inflammatory conditions like cancer and cardiac cachexia that have been extensively studied in the context of their effects on the body, but not the brain. So I think there's just an incredible opportunity to study psychiatric conditions from this new angle. All right, so let's start with the acute sickness behavior. So this is probably old news for many people. This can be very nicely studied in mice. And the basic idea is, of course, that we have pathogens that are detected by immune cells. They are specialized through pattern recognition receptors. And we don't even have to use the actual pathogens. We can just use something that mimics them. Typically, what we use is a bacterial component, lipopolysaccharide LPS, a cell wall component of gram-negative bacteria. And cells recognize this, immune cells recognize this as a bacterial invasion and produce a cytokine code. And as I said, just like in people, mice exhibit loss of appetite, social withdrawal, fatigue, altered sleep patterns, and all those other things. So this is great to have a mouse model. So this is what we did, and we is really Alita and Jun Shao, two incredible PhD students in the lab. This is a new project, and sort of they're very brave to do this in my lab. Now, so what we wanted to do is to activate the immune system in different ways. So we used 3 different toxins. What I mentioned, LPS for gram-negative bacteria, LTA that mimics gram-positive bacteria, and poly-IC, it's a viral mimicry. Then we do a battery of behavioral tests, and after 2 hours, we collect serum and analyze the cytokine patterns. And just to give you a sense, these are we use different doses sometimes, and this is the kind of pattern in cytokine responses we see. Now, is there a cytokine pattern code? So this is a correlation matrix across 19 cytokines and chemokines. In fact, this is a subset of all of our data. And about 6 or 7 dimensions are needed to get to 90% of the variance. So we performed a multiple discriminant analysis, and each dot is an animal in one condition. And as you can see on the x-axis, it's the sickness dimension. And all animals exhibited sickness behavior. I'll cover that in a moment. But all the toxins also exhibited responses along a different dimension. And this different dimension, why is it so small? This different dimension can completely distinguish between the identities and concentrations of different toxins. All right, so how do we measure motivation? So motivation sort of can be conceptualized as an energizing mental force that allows you to overcome challenges. So we did this to our little mice. We created a closed economy, so we had to really work to earn their food. So we have this device here, and initially they earn 1 pellet for each poke, and then we keep increasing the number of required pokes. And eventually the mice give up. So you can see they poke, they get 1 pellet. Then we increase it, they poke enough, they get 1 pellet, and eventually they give up. And the required poke number we call the break point, and this sort of captures their willingness to exert effort. And after 30 minutes, they can start again. All right. So first we wondered whether cytokine code can predict this willingness to work. So we use a lasso regression to use the cytokine pattern to predict this maximum breakpoint. And as you can see, it's a pretty decent prediction based on three or four cytokines typically. Maybe it's not very surprising, but we felt very good that we can actually predict one type of behavior. Now, turns out we have different behavioral metrics as well that are related to motivation. We also measure how much these animals move around, and they're mostly correlated, but there's some conditions when the animal is moving around a fair bit, but it's not willing to invest effort to get food. So there's some difference, and it turns out that we can predict these, again, based on the cytokine pattern, where the animals are overall exhibiting sort of something that the broad category of sickness behavior, but there's some differences. So this raises really the possibility that there's a cytokine code that might be different, signals something different to the brain. Because although these behaviors co-occur, I would argue that they're really sickness behaviors that might need to be invoked one at a time. All right, so how could the brain read this out? So here we are working with an ex-Cold Spring Harbor colleague of mine, Pavel Osten, using his automated pipeline that I'm not going to get into. We can sort of, what we're trying to do is to find convergently activated brain regions using brain-wide cellular resolution screens of activated neurons. And it's really exciting to sort of look around the whole brain and search for new areas. Unfortunately, I cannot report success yet. It's fairly complicated. But I do want to say that this is a very active field, and I want to draw attention to 3 really beautiful papers that have used similar strategies. One is by Asya Rolls on the insular cortex, and 2 other papers on different facets of sickness behavior that are really tour de forces. So it's kind of an exciting area now to try to really zoom in on the cell types that are controlling these different behaviors. All right, so that's it for the acute stuff. Let me quickly turn to chronic illness, so cancer cachexia. So the Greek physician Hippocrates already described cachexia over 2,000 years ago. The flesh is consumed, the shoulders, clavicles, chest, thighs melt away. This illness is fatal. And in fact, about a quarter of cancer deaths are due to cancer cachexia. About half of the cancer patients at the end stages have cachexia, but there's other conditions like heart failure that also come with cachexia. And it's a multi-syndrome disorder. Lots of things are happening, loss of muscle appetite and so on. But there's also a depression-like phenotype, extreme fatigue, apathy, lack of fatigue that further decreases the quality of life. And often this aspect of cachexia is not studied, and we don't really target treatments for this. But it's also an opportunity for neuroscientists because there's good models, mouse models for this, and that's very exciting. So for this project, we're collaborating with colleagues, again, at Cold Spring Harbor, Tobias and Miriam, and it's really led by a really impressive postdoc, Sarah Starista, in the lab, who's sort of taking on all sorts of different aspects of this project. And the basic idea, it's so wonderful to have a simple biological model for us. We inject a cell line, a carcinoma cell line, and the tumor starts growing, and the mice remain relatively healthy for a while, for a few weeks. And after that, they start losing weight and exhibit cachexia. And we terminate the experiments at 70% of the control weight. And this is what I'm going to show you. The data always align to the endpoint where we terminate the experiments. So the question is there loss of motivation? So very briefly, we use the same effort sensitivity assay. The tumor-bearing animals lose weight, and just as they lose weight, they also lose motivation to work for food. This you can see it in detail through the circadian cycle. It's really, really nice to see that there's a very nice circadian rhythmicity to the effort. Not necessarily the amount of food they get, at least in a control condition, but the effort changes. Anyhow, on the other hand, we also tested whether they prefer sugar to water, and in fact, cachectic mice drank a lot less, but they still preferred sucrose over plain water. So it's there's some specificity to this disruption. All right, so what do we do about the brain? Here we really went after the ventral striatum and the neuromodulator there, dopamine, that's known to be involved in motivation. And so we targeted the ventral striatal region for recording. We virally expressed fluorescent dopamine sensors. It's really an incredible tool pioneered by Yulong Li at IDG at Peking University, and I think one of the institutes involved here, and it's really revolutionized the way we do science with these sensors. And we're using an effort-modulated foraging task, and I'm not going to have time to get into the details. Basically, all you need to know is that water reward or any kind of reward elicits dopamine. So it allows us to monitor what's happening. So what you can see is that the tumor-bearing mice develop cachexia at some point. And there's a deficit in task performance right around the time that they start losing weight and so on. Now, dopamine is released by water rewards here. So you're seeing the time, the few seconds after the dopamine, after the water, what happens to dopamine, and this is pre-cachexia. And after cachexia, you can see that there's sort of a large reduction in dopamine level and a change in the dynamics. And this is the time course. So the last few days that we call cachexia, and before we call pre-cachexia. So they have the same tumor. Many of the things are the same, but the animal sort of is in a different metabolic and inflammatory state. So this is really nice to see that we can really read out, as actually quantitatively, the motivational changes based on changes in mesolimbic dopamine. Now, how does tumor control dopamine? That's the million-dollar question. I can't give you a full answer. But of course, we measured cytokines, and we know that IL-6 is very strongly elevated, both in the periphery and after the onset of cachexia in the brain as well. And IL-6 is, of course, very interesting as a target because there's existing inhibitors for humans to treat chronic inflammatory conditions. Now, so we use the mouse antibody to block IL-6 peripherally, and you can see that it mostly rescued the behavior. So the green is almost at the black level compared to the red. And it actually fully rescued the dopamine. So the dopamine change was essentially gone if we infuse the IL-6 antibodies early enough. So this is very, very exciting. And that's sort of where I need to finish. And so let me just sort of summarize, or instead of a summary, kind of zoom out to ask sort of a broader question. So clearly, we have a well-calibrated set of processes to keep us in balance. homeostasis. We need to keep our temperature set, how much we eat, how much energy we use, and so on. And the brain and the body have different roles that help each other, guide each other's work. So why do things go wrong? And why do things go wrong in particular ways? Not in all different ways, but they're sort of patterns. And I think the issue is that homeostasis is hard, especially when you're trying to keep multiple variables balanced. There's many stressors, both on the brain and the body, and there's many feedback loops. So it seems like chronic inflammation is a fairly common failure mode. And I want to sort of illustrate this as a last point in the language of dynamical systems. So you can have all sorts of different inducers and failures of resolution. It looks like there's many situations, many ways to get to this low-grade chronic inflammatory state and not just back to the normal state. And I think that's an organizing, I don't know, principle, but at least some organizing idea that allows us to sort of further study this problem. That was my last slide. I wanted to really thank everyone in my lab. I mentioned the people who did the work on this project and all my other wonderful members that I'm really, truly privileged to work with, and the funders, so that we can do the work. Thank you.
Thank you very much, Adam. And maybe kind of we will ask one or two of the questions that were directed more to Adam himself, and then we will just continue for the overall discussion. So I will pick up one question that is there, and it's really awesome work, and I join this time. And I assume that dopamine is coming from the VTA. Do VTA dopamine dopamine neurons express the IL-6 receptor? And do you think it's actually IL-6 is driving that? And you kind of referred to a code, so maybe you can kind of expand on this whole idea.
Yeah, I mean, the code I meant for more the acute things when there's sort of an injury and there's something to be signaled. I think in these chronic inflammatory states, it's a little different. There's sort of convergence of factors. Now, the running idea in this field is that a lot of things happen in the striatum itself, that it's a local process there where dopamine is modulated. So we don't think so, because we also measure dopamine neuron activity. So the dopamine neuron activity is itself impacted. We don't know if it's direct or indirect, but it's certainly pointing us to a neural circuit explanation, as opposed to sort of a distributed explanation with sort of local astrocytic processes changing release and so on.
So I guess it also answers a little bit the question of whether the pro-inflammatory cytokines, where are they produced? Are they produced in the brain as well?
We don't know. We don't know. I mean, it's actually very interesting. So IL-6 goes up in the periphery early and really only gets high after the onset of cachexia. And there's also blood-brain barrier disruptions then. So we don't know if the blood-brain barrier disruptions are probably indirectly, at least, if not directly, driven by IL-6, and that's how IL-6 gets in, or that's also sort of an amplification process. But we can block this with the IL-6 antibody peripherally.
And then, well, I guess it's really eventually, Michal is really missing here to put the connection there of how the peripheral IL-6 can also change the overall environment. So then maybe let's start the overall discussion. And I think what came up from both of your talks, and I would say also from Michal's talk, is this idea that we keep using this mapping, right? We keep using this mapping as a way of, you use this code, cytokine code. Carlos is using this idea of kind of an atlas representing activity. And it kind of begs this homunculus idea that there are many different homunculi, right, that represent that. So maybe, what do you think we're going to find there? Like what are we looking for, how they interact, and what do you think we're going to find there? Maybe let's take it to Carlos and then back to Adam.
Yeah, the way I think about it is that that's how science often starts, right? I mean, you first start by mapping out things and transforming one thing from one space into the other, right? From one language into the other. But it's true that often this kind of still leaves, I have this slide I use for teaching, right? From Men in Black, for the ones who are old enough to remember that, right? Where the skull open and there's an alien in there, right? And I always say, well, how many times do we need to open that? I think that we are now entering exciting, or we are already in the middle of exciting times, because we're going now beyond this homunculus idea, right? Really thinking about how is information now being used to then really activate different motor output patterns, right? Or how can we really find and explains ways using theory. That's another way people are approaching this, right? To explain maybe some computation, some higher order computation of how this is working. Now, obviously, we don't know how to do it, because if we would know how to do it, we would know how to solve the brain, right? But I think that there are these steps, right? There's the stamp collection stage, there is the atlas building stage, and then there is the dynamic stage, where you start looking at how these things change around, and then you go in and try to see if you understood the language, right? And so, in my view, this is how things progress, and we are definitely now in the stage where we're going beyond the homunculus, and I think that's exciting. But you cannot get there without making the stamp collection and the atlas part. I don't know how Adam thinks about that.
Yeah, I don't think I would call it a homunculus. I think we're just searching for the regions that we're not sort of aware of at this resolution. And I think that's just a mandatory first step before we can really understand the computations and what's happening. I also think it's just very exciting to sort of see different fields come at the same problem from very different perspectives. So when I started getting into this and trying to read about cytokines, it's just so interesting how neuroscientists would approach this versus an immunologist. And I think it's just going to be very exciting. And I wonder if immunologists might think the same thing about what we do in the brain as the very first, strange first thing to do compared to what they would have done. But I wouldn't downgrade sort of these mapping results as homunculus. It's just really the first steps.
And then it seems that motivation, it also comes up in the question, right? Motivation appears in everything there, in every possible work. And then I guess the question there is, what's going, like, why motivation changes when you show in sickness behavior, you show the motivation changes. So in one case, you can see that they are more motivated to feeding or heat or something else. So the motivations are very different. And I think it also speaks to the whole idea that if there is a code that controls motivation, what is the structure, or what should it integrate in this sense?
So Jeremy Borninger is also asking a similar question about the energy constraints of mounting an immune response. And I think it's a very related question to what you're saying, Asya. I think that's what we have a brain for, is to figure out the cost benefit kind of scenario. I don't think that's in the code. I think your sense, sort of the brain senses the periphery and the pathogens and the organs through these mechanisms. But the brain has to decide whether the sort of the cost of mounting an expensive operation outweighs the benefits. And there's not an easy answer, and I think that's why we have a brain. So I do think once we get to those kind of interesting questions, we are sort of back into systems neuroscience, where I feel really comfortable.
And maybe I think there's an additional way of thinking about it, right? Like, in case the body does its stuff because it really needs to do it, the brain is left having to deal with the consequence and find a solution of how to deal with that, right? So it's kind of the, right, because I think I mean, evolutionary speaking, for me, it's fascinating. I like to really read a lot of microbial literature, right? Like, how do microbes predict the future, right? So, because they can do that, right? So there's this fascinating study where gut microbes, when they get into, so what that happens, if you increase the temperature a little bit, they will actually start expressing pH pump, which make them acid resistant, right? And so the idea why that happens, because when they get into your mouth, they have evolved to know that in a few minutes they're going to be in a very acidic environment. So they better start actually already now expressing these predictive things, right? And a bit that's how the brain does it, right? And sometimes there's no choice, right? You need to act, and you need to, the organs need to do stuff. And that's an internal thing. Now, what the brain can do is it can change how the whole organism interacts with its environment, which obviously organs cannot do that. So it has to take this big picture decisions of how to navigate the world according to what the internal world is. So kind of the way I think about the nervous system is that it evolved to deal with a problem that individual cells within an animal are not independent enough, right? Are not independent anymore. And so now you need someone else to take these big picture decisions.
So I think it's maybe, Carlos, it will take us to this the question that keeps coming back. If you can explain protocognition, and I think from there you can also answer the question that many people ask: flies?
Yeah, well, so there's two ways of thinking about why I like to use the word protocognition, because again, I think that cognition is very often associated with really higher order cortical type of computations, right? Creativity, things like that, which obviously it is associated with, right? But, or neuroeconomic decisions, right? Should you buy the car today, or should you get a loan, or whatever, right? Now, we haven't evolved to deal with money, right? We haven't evolved to make decisions about mortgages. So where does this come from, right? So what's the what's the original building blocks to think about these computations? And as Adnan was saying, it's about dealing with limited resources, right? It's about making these trade-off decisions. And honestly, again, unicellular organisms have to make these decisions, right? Do I invest into expressing transporters to get more nutrients in, or do I invest into making a new cell, right? And so somehow I feel like many of these, and if you scale this up, obviously now into really making predictions over larger time scales, or using information which evolutionary hasn't existed, but only exists within the time life of that organism, I think then you start getting into this more brain cortical type of cognitions. But the origin of that is to deal with two resources, food and mates, right? And they're not easy, as Adam was saying, they're not easy decisions, right? So how to really deal with all these complex loops. And so I don't think you need to go very far into human cognition to start understanding the building blocks for these computations. They must exist already very early on in life. And obviously invertebrates, flies, C. elegans, I mean, bees are amazing, right? I mean, what some insects can do, or ants, is amazing. So they must have evolved early on. If not, they wouldn't be around. And so we can get insights into these things by studying these numerically simpler brains, and then get the real mechanistic insights to then look into that in humans, for example. So that's the other way of thinking about it, is that if I write cognition and flies in a grant, it won't fly. So I use protocognition to not piss off anybody. That's another way of answering that question.
That's a good one. So actually it brings us to, I think, to another level, and it's relevant very much to what Adam was talking about and what you're talking about. But many of the decisions eventually that we do, including sickness behavior, including nutrition, are completely cognitive, and they are affected by states of mind, right? Or the way you perceive cognitively a certain state, you can almost switch the system. Or to how do you think that is happening, or to what extent it's really happening? Maybe Adam?
I mean, we don't know, of course, but I think it's almost sort of a question of perspectives. I think if you're thinking about it as sort of a, what's in neuroscience called a labeled line code, where you have sort of one cue wired to one response, that sort of might not be the way to think about it. Or if you think about sickness behavior as just one entity, that may be not the way to think about it. But if you think about the process more as a new sensory system that's kind of been unexplored, that provides a rich set of information, then now you can start to think about this issue the way a lot of other sensory systems are thought about, which is, yes, there's standard transformations that happen, there's typical patterns that they can elicit, but it's context dependent, and we can kind of start to probe the conditions when sort of different ones get integrated, where there's cost-benefit analysis. To me, it's really kind of a perspective, but that, again, comes definitely from my systems neuroscience background.
Carlos?
Yeah, I totally agree with Adam. I think that one can start thinking about interoception as just another way of doing sensory information. And then you can start thinking about mapping these like in sensory space, right? Doing psychometric curves, right? I mean, when we play around with the different nutrients in the diet of the fly, you can think about it as a psychometric curve, to think about it again as a system neuroscientist, right? I think to the question you were also asking about these sudden switches in perception and like mood and so on, I think that's a fascinating question about what are internal states, something which more and more people now use. It's also system neuroscience, right? But we know that if you look at brain activity, there are these switches which happen, right, with pupil dilation and so on, with being also part of that. And so to which extent these circuit dynamics and changes are these, how they relate to internal states or not internal states, or do internal state, like, sorry, physiological states, tap into these dynamics and so on. I think that's a fascinating current discussion within systems neuroscience, which we do not understand. And I think the dialogue there for me is really fascinating, right? Like an organ state, a transcriptional state, what's the real difference to a brain state, right? So I'm a trained molecular cell biologist, right? So I like to think a bit about fundamental principles controlling these things. Is there something fundamental different about how the brain does it, or like what would be like a liquid state machine at the level of the liver, for example, right? So I think there's a lot to be discussed and discovered and identified by these discussions across different fields.
Do you think we are ready to answer those questions? You think?
I think we're ready, because the molecular tools, the cellular tools we have been using for a long time now can be actually implemented and dynamically controlled with precision within organisms. It's a high dimensional space. So what to really manipulate is the question. As Adam was saying, cataloging is really important there, right? But I think that we are ready for it. And again, I mean, you know, if you think about it, fields in biology have always been doing that. Developmental biology, right, is a systems question, right? You cannot look at the development of a single cell in a certain state, right? I mean, immunology at the whole organism level has always been asking this question. Physiology, I mean, you can argue that what we're discussing is just the 20th reincarnation of physiology as a science, right? Like 10 years ago, it was called systems biology, right? Now it's brain. It's the question of asking regulatory principles of living systems at a whole organism level, right? I think we are ready, and the progress of the field is showing that, and it's exciting moments also because of that at the moment. But it's challenging, right? And we often don't know how to do it. But you know, that's why we need talented people working. Right? And the young people nowadays are amazing.
Yeah, absolutely. And Adam is kind of took us to this, I think one of the most challenging frontiers is psychiatry, right? And Carlos also mentioned that sensory systems might be altered in psychiatric conditions. Adam, how do you think we we can approach this psychiatry in a more systemic level? Do you think it's basically, maybe it's the only way to approach psychiatry on the one hand, but on the other hand, what do you think kind of the gap that we have there in order to be able to answer that, to kind of start touching these questions?
That's a great question. But before, sort of one of the answers is that we now have the tools, which is also an answer to what the previous discussion you were just having, that yes, it might be a rehashing of what used to be called physiology, but it's not without merit, because now we can actually address things that we just couldn't before. And that's how I feel about this situation, is that for the first level, I feel like it's really immunology and other fields of biology that are providing us with models that have a psychiatric phenotype. So in that sense, it's looking in the brain of diseases where people just haven't looked in the brain yet. And again, cachexia, it's not only for cancer, right? There's a lot of other cachexias. And then we can start looking at convergences, right? So we know that chronic inflammation, even if it occurs in the context of, say, rheumatic arthritis or cancer cachexia, is very different at the bodily level. And yet they have shared behavioral phenotype. So that's a very exciting question, because it suggests a certain type of convergence. So I think that would be the sort of next step to think about this problem. And I think slowly I would hope that that takes us to clinical depression, which, to be clear, I don't think it's the same thing, but we might get a handle. And in particular, right, in clinical depression, sort of probably the brain to body arc of this many loops might be more important. But I feel like until now it's been very squishy, at least for us trying to do psychiatry. And I think this really, really strong immune component, I think, really puts us in in front of incredible opportunities.
And Carlos, how do you think nutrition, like you mentioned that before in the context, is there evidence that kind of establishes nutrition as a strong factor in this disease?
Well, I always feel uneasy to talk about that given my training and my background, but definitely, I mean, there is a strong movement in so-called nutritional psychiatry. There is obviously a lot of interest in the association between microbiome, brain, and psychiatric diseases, thinking about the microbiome as a nutrient producing and modifying entity, right? Puts it maybe in that direction. Now, if you talk to like practicing clinicians, right, many of them are doubtful to which extent these things are really reproducible and impactful. They're worried also that, this opens the Pandora's box to charlatans and also to people avoiding efficient treatment. So I think there's a promise. There is likely to be a biological background to it, but how to really implement that in clinical practice, how to do that in a very targeted way, because it's clear that it has to be personalized also, right? And to which extent it is treating epiphenomena, right? So, for example, most children with autism have digestive nutritional challenges. And so diet interventions are strongly associated with an improvement in well-being and in cognitive performance of these children. Now, are you just making them not feel sick and that actually helps then the brain? Or are you directly intervening at the brain level? We just don't know, right? And ultimately, right, again, think about brain-body interactions as a whole system. Does this really matter? Right? But it is a promising, complex question. And my personal opinion to this is the way forward is mechanisms, understanding how it really works, so that you have then a more objective molecular platform to then evaluate some of these things beyond the clinical epidemiological approach, which is obviously very important too. But doing prevention is even more complicated, right? Because in nutritional interventions over 10 years, that's hugely challenging.
So actually, in a way, maybe it all brings us to a question that I think is science, we always look for causality. Maybe it's a way our enemy in this case in brain-body interactions.
I'm not sure I understand your question, Asia.
The way we usually look for causality, that something, this is the initiator and this is the outcome. But when we are talking about these systemic systems, maybe the fact that we are looking for causality, we're actually shooting our own leg. Maybe we're doing the wrong thing here. Maybe we need a different paradigm to study brain-body interactions because causality is not unidirectional anymore.
Yeah, okay, I think another way to ask that maybe is what's the limit between reductionism and systemic thinking about that, right? And I think that's also where I feel I've learned a lot from thinking about from system neuroscientists, right? Where at one stage you have to go okay, let's try to model that to start seeing things which maybe you cannot put so easy into a narrative perspective, right? And start using more modeling approaches to start understanding that. The question is obviously if the model is correct, but you cannot make a narrative out of it, is this really understanding? But then we are really going into a philosophy of science type of discussion, right? But I think there is value of using computational models when thinking about multi-dimensional, multi-feedback loop type of systems, which I think we can learn a lot from. I personally learned a lot from system neuroscientists thinking about that. For my colleagues here, Jean-Paul, for example, who do that. Yeah.
So then maybe in the last 30 seconds that we have, Adam, do you think in the concept of neuroimmunology, where there was the idea that immune cells don't enter the brain, is there something like that that holds back the whole field for you?
I don't know if it holds it back, but I think when I was looking at the literature of this sickness behavior type, I think there's sort of no circuits in it. It's all cells across the body and the brain, but no circuits. So I think the new insight, and that's not new to a neuroscientist, is that the answer may be in sort of cell types connected to other cell types that we call circuits.
So then I think we will wrap it up, and maybe we will wrap it up. But by the way, we started the whole talk together with Limor in the beginning, when we were talking about between ourselves, who is in the brain-body field. And I think it's really everyone. And I think this is the most exciting part, that everyone are part of this field that basically needs all this perspective to be formulated. So with that, I would like to thank you very much for your participation.